Our experiment is going to demonstrate the surface tension in plants using colored dye. The colored dye will show us where the water travels and where surface tension occurs. This will prove to us the importance of hydrogen bonding in water and how the surface tension of water plays an important role in holding the flower petals and the stem up. We're going to do this experiment by demonstrating one carnation flower undergoing hydration and experiencing intermolecular forces and hydrogen bonding within its petals. We will compare this to a carnation flower undergoing dehydration and therefore experiencing a lack of surface tension. This dyeing process works best with warm water because of the fact that heat acts as a catalyst. However, if the water is too hot, then it could possibly result to an error in this experiment because if the hydrogen bonds within the water are disrupted, then its surface tension will then decrease. Different colored dyes seem to work at different rates due to the chemical makeup of the dye, while different brands of food coloring also affect the rate of color absorption. Experiments indicate that blue dye is most effective, followed by red dye and then green dye. So because of this, we're going to use blue dye in this experiment. As the colored water is absorbed, we will be able to see exactly where it supports the flower structure by its change in color.
Within the hydrated carnation, as the water molecule evaporates from a leaf, it will pull on every other water molecule in a long chain down and through the entire plant stem and due to hydrogen bonding. Hydrogen bonding plays a crucial role in many biological and chemical processes and is very important in the water transport system and support system of plants. Within a single vessel, water molecules cohesively hydrogen bond not only to each other but also to the cellulose chain, which compromises the wall of plant cells. A straw-like capillary tube is then formed, which allows plants to pull water up. Hydrogen bonding can create a long chain of water molecules which can overcome the force of gravity and travel up the high altitudes of leaves. This amazingly creates enough pressure in the plant to hold its structure upright and not wilt.
Okay, so for this experiment, we will have one glass for the flower that will be under hydration, and then we will have this glass for the flower that is under dehydration. So this one will have no water in it. This one will have water in it. So let me add some water. Okay, and then we're going to use blue dye because it has been shown to show the best results, so I'm going to trust that. So let's see. I'm going to use some of this dye for frosting, and I'm also going to use some of the normal liquid food coloring. This is like a gel, just to get the best results. Okay, so I kind of want a deeper blue than that. So I put about 13 drops in there. Mix it up again. And it looks pretty dark. So what we're going to do now is we're going to cut about 4 inches from the bottom to the top so we can get a fresh cut. And I'm going to go in an angle so that no air bubbles or anything can affect wilting or air in this experiment. So that looks like a pretty clean cut, so I'm going to put it in. And this can take hours, it can take a couple days until we start seeing the color and where the surface tension goes, but we will check on it in a little bit and see how it's doing. And then for this one, I'm just going to put it in the same upright angle, and we can compare and contrast the two. In further biological and chemical detail, when a cut flower is placed in dye, the dye is pulled up the stem and absorbed along with the water. During a plant process called transpiration, water is evaporated from the leaves and the dye is left behind on the petals, so the majority of water inside a plant is eventually lost through transpiration. The loss of water through the leaves creates low pressure in the leaves, which causes the plant to pull more water up through its stem. Once the water begins to travel up the plant's vessel system, cohesion caused by hydrogen bonding causes the water molecules to stick together and therefore continues to travel up the stem. It will then reach the flower's leaves and petals, creating a surface tension and pressure that allows it to stay standing upright. The carnation flower that is undergoing dehydration is wilting because water is moving out of the plant cells and the pressure and surface tension is decreasing.
The extraordinary stickiness of water is due to the two hydrogen atoms which are arranged on one side of the molecule in a state known as hydrogen bonding. The fact that water can create surface tension through hydrogen bonding gives nature the proper sources to grow and develop in unlimited ways. Water moves through the plant by capillary action, which occurs when cohesion and surface tension bind a liquid together and the forces that attract the bounded liquid to another surface are greater than the force of gravity. The intermolecular forces in hydrogen bonding that occur in water allows for the cohesive properties of the water. Through these binding and surface forces, the plant stem is able to suck up its water like we do when we drink through straws. Pressure also plays an important role in keeping flower stems and petals lifted. When water moves into a plant cell, the cell expands much like a balloon does when it's inflated. However, a balloon would burst when it has exceeded its maximum space for gas, but plant cells do not burst with too much water. This is because their cell walls limit the amount of water that can move into plant cells due to the pressure within their cell wall. Pressure is one of the key concepts that keep plant petals, leaves, and stems lifted and healthy. Another one is that the surface tension within the carnation petals is the result from the very strong attraction of water molecules known as hydrogen bonds. The high surface tension of water allows it to support objects that are denser than itself. Hydrogen bonds provide higher surface tension to a liquid, which allows the plant to hold pressure within its stem, leaves, and petals. So our main educational goals that we hope you got from this are that the surface tension in plants was proven through our experiment by demonstrating the hydration of one carnation and the dehydration of another carnation, and leaving us to compare and contrast the two. Another would be the carnation and the dehydration wilted due to the fact that the intermolecular forces, cohesion, and surface tension of water were not present. And lastly, the hydrated carnation remained standing, and the colored dye proved to us that the water molecules stayed stuck together as they traveled to the carnation petals, and they created pressure and surface tension through hydrogen bonds to maintain its standing position.